Hello students, welcome back to Principles of Organismal Biology. Last class we learned that the genetic material is in the chromosomes and in particular in the double-stranded DNA. We also learned about the genetic structure, the DNA structure. This class we're going to keep learning about it, and we're going to learn more about the genes and gene expression. I hope that you enjoy this class. You probably have heard about the human genome. The first draft was published in 2001. And now we know that we have a lot of genes encoded in this DNA. But how come do we pass from having these sequences in the DNA to express it into proteins? That is what we refer to the gene expression, and that's what we're going to learn in this class. So the learning objectives today are to understand the central dogma, which was postulated by Francis Crick, to discuss and distinguish between transcription and translation, and also to know the evidence for the evolution of the genetic code. Now, how genetic information gets expressed? Let's remember what we define as the phenotype, which is all of these observable traits in an organism. So look at yourself in the mirror and find your phenotype. Now, also remember that the genotype is this genetic code, and it's the information, the genetic information that determines your phenotype, how you look, the observable traits that you have. Sometimes a similar phenotype, let's say the color of a flower, might have different genotype. So it is important to look at the genetic variation that have or confer that phenotype. The central dogma of molecular biology was proposed first by Francis Crick in 1957. Yes, this is the same guy who described that the DNA had this double helix, along with Watson and Wilkins. Okay, this dogma explains the flow of the genetic information in all the biological systems. We have to understand that the genes are in the DNA and they carry this information for the traits. Now, we pass from the DNA to produce polypeptides. So the DNA carries information that synthesizes polypeptides, and polypeptides are a change of amino acids. So build amino acid, one amino acid here, and another here, etc. A group of polypeptides form proteins. The central dogma, it's in a simplified way, includes two stages, transcription and translation. But we also have to remember replication, which is when the DNA copies itself. Now, let's think about this, transcription and translation. If you say, I love my tool for a class, and you want to transcribe it, maybe you have something kind of illegible, and then you do this, it's the same language, you just transcribe it. You're using the same language. And then from that, you wanted to translate it into a new language. I speak Spanish, I'm Mexican, so that will be, the translation will be, amo mi clase 204. I hope that you do. Well, it's the same thing when we talk about this transcription in biology. So we are using the language of nucleotides that passes from DNA into RNA. And then in translation, we convert RNA into amino acids, and that is a different language, the language of amino acids. Okay, remember the difference between DNA and RNA? Well, in DNA, we are going to be using thymine, but in RNA, we use uracil instead of thymine. The other difference is going to be that the sugars in DNA are the oxyribose, as shown in here. And in RNA, this hydroxyl group is in here, and that is a ribose. So RNA is going to bridge the gap between DNA and proteins, or polypeptides, as I have explained before. And this is what the central dogma of Francis Crick explains: the genetic, how the genetic information gets translated into proteins using this intermediary. In eukaryotes, that means species that have a nucleus, like this one, the transcription, that means the transformation from DNA into RNA, happens inside of the nucleus. And the translation is going to happen in the cytoplasm. That means outside of the nucleus. Now let's look a little bit more in detail about what happens in these stages. During transcription, we're going to have this DNA, this double-stranded DNA, it's going to be open. So the two strands get separated, and now one strand functions as a template. This template is going to be the scaffold for the formation or the synthesis of RNA. This RNA is called messenger RNA, sometimes notated as mRNA. Now, here we have this pre-messenger RNA because during this, before exiting the nucleus, it's gonna be it's gonna get modified. Okay, once that it matures, when the modifications are done and this messenger RNA is ready to exit the nucleus, it exits and to the cytoplasm. In there is gonna be read by the ribosome. This is the ribosome. The ribosome is going to be made out of ribosomal RNA, right here, ribosomal RNA, and also proteins. So this looks like a hamburger to me, and this hamburger is going to be reading the messenger RNA, this information. Now, if you look carefully in this RNA molecule, we have a sequence. And this sequence now is going to serve as a scaffold to form the polypeptides. In order to form the polypeptides or to synthesize them, we need this transfer RNA. This transfer RNA is going to carry an amino acid. This is the amino acid that is being attached by the amino acid tRNA synthase. Once the messenger is read, then the ribosome allows that particular tRNA to be attached, and then it reads the next three nucleotides. And if, depending on the information, it would allow to attach the proper amino acid in this change that is forming the polypeptide. The product of the gene expression are polypeptides, and these polypeptides form proteins. Some proteins are enzymes, and that means that they have a biological catalysis. That means that they can accelerate some biological processes. For example, if you think now in your favorite food, maybe you can think in a hamburger or ice cream. Yes, let's think about ice cream. If you close your eyes and start thinking about the ice cream, you probably start salivating. And in this saliva, you have amylases. These amylases stimulate digestion, and they are going to break down the starch in your food. So you are starting to get ready for digestion when you're thinking about that. Isn't it cool? Okay, so many proteins can be formed by several polypeptides, or just a few. And in some cases, we have examples of polypeptides or proteins that have a very important function. For example, tyrosinase. This enzyme lives in the chromosome 11, right here. This enzyme is going to be responsible for, it's associated to the pigmentation. If we have a mutation in this tyrosinase, we can have the formation of albino. Now, let's watch a little bit more. Let's watch this animation to learn more about transcription.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
What will be the RNA sequence of this DNA segment after transcribing the upper strand, right? Because the transcription goes from 5' to 3'. Okay, I hope that that was very easy for you. As we have mentioned before, RNA doesn't have a thymine. Instead, thymine is being replaced by uracil. So basically, you probably you could have used this strand as a template. So if you use this template strand, then the complementary of A will be U, and then G, G, U, 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 G, G, C, U, C, A. So the answer is B. This is the transcribed messenger RNA. And let's say that we have this, this is a molecule of DNA, and this sequence is part of this gene number 3. So just for you to visualize a little bit more where this sequence will live in the DNA. Now, when we have a translation, then we're going to convert this messenger RNA into a sequence of amino acids that we call polypeptides. So UGG has the information to translate this into a tryptophan, an amino acid called tryptophan. UUU has information for phenylalanine. GGC, glycine, UCA, serine. We are going to learn about the genetic code. Now, you probably are thinking, what are the instructions for assembling amino acids into proteins embedded or encoded into the DNA? And this was a question that tortured a lot of researchers because we have 20 amino acids and we only have 4 nucleotide bases in the DNA, if you remember, A, T, C, and G. So how many nucleotides correspond to an amino acid? This is the genetic code, which consists of 64 triplets of nucleotides. And these triplets are called codons. With the exception of 3 codons, each of these are going to codify for an amino acid. So these exceptions are right here. And these are signals. These triplets are a signal for the ribosome to stop translating. We also have a codon, AUG, that is a start codon. So that when the ribosome is reading, it starts with this AUG. When the ribosome starts reading an AUG, it knows that it's the start codon, and the methionine is getting added into the polypeptide. So how do we read this table? So the first mRNA, the first base in this messenger RNA is in this site. So you will have U, C, A, and G. The second base in this molecule will be over here, and the third will be over here. So let's find an AAA. So we'll go over here, A, and then if we go to the second base is right here. So let's intersect it, and the third will be over here, A, this A. So AAA is right here, and that codifies for a lysine. The genetic code is redundant or degenerated. What that means is that we're going to have several codons codifying the same amino acid. In this case, for example, leucine, it's codified by 6 different triplets or 6 different codons. In comparison, phenylalanine is only codified by 2 codons. So that's redundant or degenerated. I have a question for you. Leucine is the form most abundant of all amino acids in human muscles. How many codons codify for leucine? Okay, I think we have mentioned it before. That is 6, and we have them right here. Another question. What will be the amino acid sequence of AUG, UCU, UU, GG? So the first thing that you have to do is to split this in triplets. So AUG, where is AUG? We said before it was a methionine here. The next one is UCU. Where is UCU? Let's go find that. UCU. UCU. The next one is serine, right? So methionine, serine, and the next one will be phenyl and glycine if you keep reading this. So it's very simple. So the right answer is E. The genetic code, the reading of the genetic code when we are reading the messenger RNA needs proper translation. And that means that it depends on the correct reading frame. Let's think about this. The red dog ate the bug. The red dog ate the bug. That is a triplet, right? And it means something to us. What if we shift the reading frame one letter? That will read something like the herd ed ox, ted hep og. That doesn't mean anything to us, right? And if we shifted 2 letters, it would be something like the do gat et ebu g. That doesn't mean anything either. Same thing happens with the messenger RNA. It needs to be read in the proper reading frame in order to assemble the right sequence of amino acids. Okay. The genetic code is universal, and that means that different species share the same information, or they are being read in the same fashion. That means that AUG, for example, is gonna have a methionine in humans, in chimps, and in plants. You see, it's shared universally. And another example is CCC, always codes for amino acid proline. And this is very interesting because Now we can use that information to synthesize some products. For example, we can use human genes. This human gene that has been isolated can be inserted in a plasmid. And a plasmid is a circular bacterial DNA. And if we insert it into the plasmid inside of the bacteria, now the bacteria can synthesize this product. In this case, it can be insulin. And we could use it as a drug to for people that need to have injected insulin. So that's very clever. Okay, so today we learned about genetic expression and the genetic code and central dogma. I hope that you have enjoyed this class. Thank you.